Right. So this is chapter 9. We are looking at Mendel and gene idea. Like I said, I'll be going straight to the things I want to emphasize on. Um, so when Mendel was going to study the concept of gene, he used pea plants. And the reason he used pea plants is part of it is so that he can study many generations. He couldn't use animal, of course, because think about it, take 9 months to give birth. The time for the individual to grow, reach puberty. Now, he himself is not going to be alive to witness many generations, so he can't study using human. So he had to use pea plant because they regenerate quickly, and then he is able to see successive generation. Um, all right. Now, if you so when he did it, the question is that how are traits transmitted from parent to offspring, right? That was the question, and the answer to that question came out to be gene. All right. So the question that led to discovery of gene is the curiosity that how is it that parents transmit some traits and you see some traits in the parent in their offspring. You see it in their children. That how does that happen? That means there's something responsible for it. Um, so it was that question that led to the study, which is what now provided the answer, which the answer is gene. Now, so how are traits transmitted from parent to offspring? So this parent is purple, and this parent that is purple, of course, this parent here, we say this parent have two alleles. Now, what is allele? Alleles are pair of genes. Alleles are pair of genes. Alleles are pair of gene, which is what you see here. I mean, I will annotate it now. Give me a moment. So this is allele. They are pair of genes. Now, when you say a pair of gene, it means that even though this is, let's say this is the father now. This is the father in this case. Now, this is the father. But this father also have parents. And you expect that the father will also have mother and father. So this father, of course, will have gotten genes from one from his own father, another from his own mother. So for every trait that you have inside of you, there is a contribution from your father and a contribution from your mother. That is the idea. The same thing when you get to the mother here, this mother as well have this trait from his father, from her father, and this from her mother. So now, that is the idea of traits. Every gene that anybody have, there is one from father, there is one from mother. So that is why the allele is a pair of gene. So now if you are looking at my height now, I have a gene that is for height from my father. I have a gene that is from height from my mother. All right. So that is the idea of alleles. That's why alleles will be two. Every gene have two copies. Now, interestingly, if my father is fair in complexion and my mother is also fair in complexion, hypothetically, then I'm going to have both genes from them that is coding for the same color. But it is possible that they are different colors, and my father can give me a gene for light skin, and my mother can also give me a gene for dark skin. In that case, it will now be a function of which of the gene is going to be dominant gene that will be expressed. The gene that you see on my outside is the gene that control what you see on my outside is the dominant gene. So I'll be looking at all of that in a bit. But I've told you alleles means pair of gene. And why is it pair? It's because one is coming from father and the other comes from mother. So now, when these parents want to give birth to a child, you see this allele, of course, we did mitosis and meiosis already, right? So this allele, these parents will go through meiosis, which is what will form gamete formation, right? And then sperm will be produced. So sperm one, two, three, four. And of course, you know that all these sperms have different genetic variation because they are different and they are haploid, your knowledge of meiosis. And the same thing here, this woman as well will also give rise to different haploid cells. And you remember, of course, in females, only one of the egg that is produced, only one of the egg, only one egg out of the four products is functional. The other, we call it polar bodies. Don't mind me, I'm writing with my mouse. All right, so the polar bodies are the other three. One egg is actually functional egg. Now, so you see the two alleles have to separate, which is what happens here during meiosis, gamete formation. Now, fertilization is going to happen. So these two guys, the sperm fertilize the egg, and then a new organism is formed, which is what we call the zygote. At this point now, this is a new organism coming from one from this parent, another from this parent, which is what happens to every one of us. And this is not just for anything, this is for every trait. You have a trait from your father, a trait from your mother. And then when this individual is also going to have a child now, want to produce egg or sperm, this individual also will go through meiosis. When it go to meiosis, which is gamete formation, all right, I'm going to write meiosis here. Gamete formation is meiosis. All right. Then this will produce four different organisms as well. I mean, four different haploid cells as well. Now, this is the basis of variation that we talk about. Now, this tells that there's something that organisms transfer to their offsprings. Let's go. So Mendel discovered the basic principle of heredity by breeding garden peas in carefully planned experiments. So Mendel's first approach to study heredity allowed him to deduce principles that remained elusive to others. A heritable feature that varies among individuals, we call it character. So your height, so when in biology now, when we say character, it means those are features that are inherited, and vary among organisms. All right. Each variant for a character, such as purple or white colors for flower, is called a trait. So sometimes we use character or traits interchangeably, but the character, the feature itself, like the color itself, is the character. Now, the variations of the color is what we call traits. So height is a character. The variation of the height is traits. So you can say somebody is tall, that's a trait. Somebody is short, that's a trait. But height itself is character. So it's like you're saying these are the characters, but they can have different roles. All right. So peas were available to Mendel in many different varieties, so you could use them. Other advantages is that they have short generational time, like I said earlier, large number of offspring, mating could be controlled. Plants could be allowed to self-pollinate or they could cross-pollinate. All right. So Mendel chose to track those characters that are called in two distinct alternative forms, meaning that they can have two traits or two forms, right? So he also stated, like now we can talk about purple flower and white flowers, right? Now he also started with varieties that were true breeding. Now, what are true breeding plants? They are plants that produce offspring of the same variety when they self-pollinate. I will explain that to you. Now, this is these plants. check something. All right. So when you say plants are true breeding, it means that, like you see here, they will produce uh offspring of the same variety when you self-pollinate them. So if you look at this now, this is true breeding. This plant, if it pollinates itself, it's going to be producing the same version of itself. Purple pollinate purple will give rise to purple. Now, what that means is this: they are going to give rise to the same phenotype. That means usually they will not be different, they will be the same. All right, I'll talk more about that as we go. So in a typical experiment, Mendel mated two contrasting true breeding varieties. Now, what does that mean? He mated two contrasting true breeding. So this is true breeding. This is also true breeding. He mated the two of them together, purple and white. All right. Now, the true breeding parents are called P generation, and the hybrid offspring of the P generations are called F1 generation. Now, this is what my breed. When F1 individuals self-pollinate or cross-pollinate with other F1 hybrids, the F2 generation is produced. So let's go. This is what it did in actual sense. Now, this is what we call the true breeding plants. Now, the first thing about true breeding plants is that when you pollinate them, they are going to give rise to the same phenotype as them. So if you look at this now, this is the purple flower. So this Mendel cross-pollinated the purple flower and the white flower. Now let's fix this as, so let's take this as purple. Let's use P, uppercase P for purple, PP. Then let's use lowercase p for white. Now, by the time they cross-pollinate this, what is going to happen? These guys will be uppercase P. If you don't understand this yet, not a problem, I will explain. These guys will be uppercase P and lowercase p. Now, but the fact that they are uppercase P and lowercase p means that this purple is dominant. And since purple is dominant, that means all of the offspring will be purple. Now, they will be purple in appearance, in their phenotype. Phenotype is physical trait, physical appearance. In their phenotype, they are purple. But when you look at their inside, they have both purple and white. So genotype, which is genetic, it's purple and white, uppercase P and lowercase p. So we say that these guys are heterozygous. Okay, I'm going to use this. We say these guys are heterozygous. And we say that these guys up here, both of them are homozygous. Homozygous because they have the same lowercase p, and the one that is purple also have the same uppercase p. So they have the same gene. Now, don't forget, this is allele. So homozygous allele, and this is heterozygous allele because it is having different gene. So it's like the father gave gene for high color that is different, brown, and the mother gave blue. Now, both of them have different eye color. That's heterozygous. Heterozygous means different. Homozygous means the same. So this child now, we call this the parent generation, right? We call this the parent generation, and we call this F1 generation. F1 means first filial generation. F1 means first filial generation. P generation means parent generation. Parent generation. And then the F2, which will be here, I will explain that later. It means second filial generation. All right. Now, so in the first filial generation, all the plants are purple. Why? Because the P is the dominant trait. the dominant gene, and the lowercase p is a recessive gene. The gene that is expressed is the dominant gene. That's the gene that affects the phenotype. So even though in the genotype there is, so like look at this now, I'll write something here. For this F1 generation now, genotype is both lowercase p, uppercase p, and lowercase p. That's the genotype. They are both purple and white when you look at their inside. But the phenotype means physical appearance. They are all purple when you are looking at them. So when you're looking at them with your naked eyes, they are purple. They don't have any whites. But when you go to study their genotype, you realize that they have both purple and white in their genotype. Now, but when you get to F2, what's going to happen? In F2, you notice that this now there are some in number. This is 705 purple, and this is 224 white. You know what happened? All that happened here is that they cross uppercase P against uppercase P and lowercase p. They cross both of them. And so I'm going to try to see if I can do crossing here so that I can start to show you what we are talking about right away. Now, on this, you can use crossing and you can use Punnett square. If you know how to use Punnett square, then that helps. I will still show you Punnett square. I can't do Punnett square here. I can do it anyway, but I will still show it to you since it's there. So from here, what you have here is this that produce this purple. This will separate first, and if you separate them first, there's going to be P here and there's going to be P here. The first thing that happened is that the genotype must separate first to form gametes. So we call this set, this is a genotype, these are gametes. So this is a gamete, this is a gamete, this is a gamete, this is a gamete, but this is a genotype. You don't transmit a genotype, so it must separate first. Sometimes when I'm doing it because I'm fast, I just want to quickly do it, but I don't want to forget it's a law. We call it law of segregation. These must separate first to form gametes before it can be transmitted to an offspring. So the next thing we do is crossing. We take the first one, take it to the inner one here. That's going to give us uppercase P and lowercase p. We take the same first one, take it to the outer. It's going to be uppercase P or uppercase P and lowercase p. We take the inner one here, take it to the inner one here. Then we are going to have the same uppercase P and uppercase P. I'll just make it this one. Then we take the inner one here to the outer one here. We have uppercase P and lowercase p. And now what you notice is that all of the offspring in the F1 generation are all the same, and they are all heterozygous, and they are also all purple. So we call them heterozygous purple. All right. Now these guys, now look at this. These guys are the homozygous purple because they have purple, all true, pure purple in their gene. But these guys, they are heterozygous purple. We say they are heterozygous purple to just differentiate that they are not the actual pure purple, they are heterozygous purple. Now, if you pick these guys now, one of them, two of them, and cross it, let's say one of them is fertilizing the other one, which is what we have here. So two members of F1 generation, if you cross it again, you are now going to have, what you are going to have is this. So let's see, I'll do this just to let you see what it looks like. So the first thing we are going to do is to separate the genotype so that we can now have gametes. So these are gametes, these are genotype. So if we cross it now, 1, this, we are going to have uppercase P and uppercase P. So if you go from outer to outer, you're going to have uppercase P and lowercase p. Inner to inner, that will be uppercase P. I will just arrange it in the same order, okay? So we could do lowercase p and uppercase P. It doesn't make any difference. That you say lowercase p, uppercase P, lowercase p, it's all the same. So the last one will be this, and then this will be lowercase p and lowercase p. Now, what do you notice here? You notice that this one is, of course, homozygous purple. This is going to be heterozygous purple. This is going to be heterozygous purple, and this is homozygous white. Now, how many purple are there? Out of the 4 offspring, you have 3 purple. So 3 purple out of the 4 offsprings, 3 purples, and just 1 white. Now, what does that mean? It means it is 3:1. So it means the ratio that determines the phenotype, this is phenotype, so we call it phenotypic ratio. It is 3:1. So, and that's what you also saw here. If you divide 705 divided by 224, you will notice that you'll be around 2.9, 2 point something. That is actually close to 3. So it's 705 divided by 224. That gives you, as a matter of fact, it gives you 3.14. So that gives you 3:1. It means that when you have 3 purple, you are going to have about 1 white when this crossing is done. All right. So in the F2 generation, you will always expect that when you cross, now listen, take note of this. When you cross the parent generation that are true breeding against each other, in the first filial generation, you are going to have all of them heterozygous, and they will be purple, depending on what we are looking at. So it will be heterozygous. All of them will be heterozygous, and they will all follow the dominant traits. But when you now take that F1 generation and cross them against each other, the F2 generation is going to be in ratio 3:1. There will be 3 purple, 1 white. Phenotypic ratio. Genotypic ratio, genotype wise, you see there's 1 purple, there's 2 of this, and there's 1 of white. So genotypic ratio is 1:1. I'm going to type that here so that you pick it well. For this, I'm talking of it now for this generation 2, or anytime you are crossing these heterozygous against each other. So genotypic ratio, genotypic is from genotype, right? Genotypic ratio is 1:2:1. Then phenotypic ratio is 3:1. So that's the standard. You don't want to forget this. This is something you should expect at every time. I'll continue now. All right. This, the same thing we've just done. I think I'll just continue. Now, Mendel reasoned that only the purple flower factor was affecting the flower color in the F1 hybrid. F1 hybrid means the heterozygous purple, the uppercase P and lowercase p, F1, first filial generation. Mendel then called the purple flower color a dominant trait. Why? Because that's what is determining the color. When both of them are present, this white is silenced. So it means the blue, the purple, is the dominant. And then they call the white the recessive. So the one that is usually speaking out, that is usually pronounced, is the dominant. and the one that is quiet, that is silenced, that is masked, we call it the recessive. So the factor for white flower was not diluted or destroyed because it reappeared in the F2 generation. Does that make sense? Mendel observed the same pattern of inheritance in 6 other pea plant characters, each represented by 2 traits. So what Mendel called a heritable factor is what we now call gene. So now I can easily say the gene for white color, the gene for purple color, but at that time they don't know what it is. And that was what he figured out. So these are the traits that Mendel did for. He did for color, he did for the seed color, the flower color, the seed color, the seed shape, the pod color, the pod shape, flower positioning, stem length. Those are the things that he did for. And if you look at this ratio for phenotypic ratios, you will see that they all look like close. So you see when you look at this is about what we did the other time. If you look at these as well, this 3.15 to ratio 1, this 3.1 to ratio 1, 2.96 to ratio 1, 2.82 approximately 3 as well, 2.95 approximately 3, 3.14 approximately 3, 2.84 approximately 3. So all of them were consistently showing about the same variation, 3 ratio 1, 3 ratio 1, 3 ratio 1. Some are just a little above 3, some are below 3, but it's still all rounded up to 3, right? So that was what Mendel realized in all the traits that he did for, which are about 8, right? Now, Mendel then developed a model to explain the 3 ratio 1 inheritance pattern that he observed in F2 offsprings. 4 related concepts make up this model, and the concept can be related to what we now know about genes and chromosomes. Number 1, he said alternative versions of gene account for variations in inherited characters. Alternative versions of gene, which is what I call alleles. And I told you that alleles have 2 versions, one from father, one from mother. So it is alternative versions of gene. So that was what he came up with. Okay, that means the fact that there's purple and there's white, that is actually what accounts for variation in inherited pattern. That's number 1. So for example, a gene for flower color in pea plants exists in 2 versions. There's a purple and there's a white. So that's where he came up with alternative version. And if you remember, when I was talking about that 2 slides ago, I talked about homozygous and heterozygous. So he's saying now that when you can have heterozygous gene, then this is based on heterozygous gene that were crossed. That based on heterozygous gene, because we have 2 copies of a gene, 2 versions, not just 2 copies, 2 alternative versions. There's purple, there's white, there's tall, there's short, there's brown high, there's purple high, different versions of a gene. That's part of the reason that is responsible for differences in us, in us when you are created or when you are born. You have 4 children from the same parent. Let's say all of them are males and none of them look alike. It's because there is variation. Now, this alternate version of genes, you see, we call it alleles. Alleles. The alternative version of genes are called alleles. Alleles. Each gene resides at a specific location on a chromosome, which is what we call locus. The gene at a particular location is called locus. The gene at a particular location is called locus. Gene at a particular location, locus on the chromosome. Let's go. Now, here is a picture of what we are talking about. Now you have one allele resulting in sufficient pigment or purple. So this is the allele for purple. You see that here on a chromosome. This is a chromosome. Now, the location of the allele on this chromosome is what we call locus. Now, you see, there's now another gene here on this same locus, but on a different chromosome. So let's say this locus, we call it number 1. This is also locus 1, but on a different chromosome. This is the chromosome from father. This is the chromosome from mother. So that means for every chromosome, the chromosome that have, so I think I'll tell you this now. Oh yeah, I can tell you now. You remember when we talk about homologous chromosome in chapter 13, when we're looking at meiosis, homologous chromosome, this is what it means. Homologous chromosome means a chromosome that have the same size, it has the same, it's on the same location, and it has the same gene on those locations. That is homologous chromosome. So these are homologous chromosome of each other, because this is this same chromosome from the father. This is this same chromosome. So let's say this chromosome is called A. This also is A. The only thing is that this A is from father, this A is from mother. So the location of their genes will always be the same location, but the gene can now be different versions, but they are the same gene. So this gene is coding for color now. This can code for purple color. This can code for white color. If you are considering ourselves, this can code for light skin. This can code for dark skin. That is what it is. So this gene, we call them, this chromosome, we call them homologous chromosome. And the genes, you are going to say this one, let's say this one is P, like we used. This one is lowercase p. And now it's going to become PP when we bring it together. All right. Now, again, I have told you already, DNA, a sequence of DNA is what we call gene. A sequence of DNA that code for a particular trait is what we call gene, right? All right, let's go. Now, the second thing he came up with is that for each character, an organism inherits 2 alleles, one from each parent. So it's more like a follow-up. I've already explained that every parent gives one, and then, so like what you saw here now, this is from father, this is from mother. Now it's going to be 2. All right. The 2 is called allele. 1 is called gene. So Mendel made this deduction without knowing about chromosome yet at that time. The 2 alleles at a particular locus may be identical. When it is identical, you remember I told you we call it homozygous allele. When it is different, we call it heterozygous allele. So the 2 alleles at a particular locus may be identical, as in what you see in true breeding, and it may be different, which in case we call it heterozygous at that time. Now, third, he now decided to say if the 2 alleles at the locus differ, then that means one of them have to be the one that is the boss. So the one that is the boss is the dominant, and then is the one that's going to determine your phenotype. And then the other one is the recessive, which is likely not going to have any effect on your phenotype. All right, so the F1, which we saw at purple flower, which is the dominant, and the white flower is the recessive. Fourth, which is what I stressed earlier, it says the law of segregation. And the law of segregation is simple. The law of segregation is that what we call this, if you say this now, for instance, if you say P, this is a genotype. You don't give a genotype to a child, to an offspring. What you give to an offspring is gametes. So these have to be splitted first. This must separate first to what we call gamete. Now, this is a gamete or a gamete. Now, this is a gamete. The gamete is what is going to be given to an offspring. So you see the two allele. This is genotype, is also called allele. Now, the two alleles for heritable character separate first during gamete formation. You see that gamete formation and end up in different gametes. All right. Thus, an egg or sperm gets only one of the two alleles that are present in the organism. This segregation of allele corresponds to the distribution of homologous chromosome, which you saw in meiosis. All right, let's go. The model accounts for the 3:1 ratio observed in the F2 generation. Possible combination of sperm and egg can be shown using Punnett square. So Punnett square and test crossing give you the same thing. The one I've done before is test crossing. You will see Punnett square. It gives you the same thing. There's no difference. You can decide to do either of the two when you need to use it to solve a problem. A capital letter represents dominant allele and lowercase represent as recessive allele. Now, there's no time when you are going to say you want to decide to do otherwise. There's no time. Dominant allele will always be uppercase, and recessive allele will always be lowercase. There's never any time when it's going to be alternated. You must use uppercase for dominant, lowercase for recessive at any time. Because even though you may feel otherwise in your mind, but if we are reading something, anytime you see uppercase, it means that's what is the dominant trait, and the lowercase is the recessive trait. So anytime you see that, just let that come to your mind straight. So again, this is what we did earlier. This is the parent generation, and I told you these are true breeding plants. Then they crossed on each other, and then now you have heterozygous dominant or heterozygous purple. Now, by the time you come here, all the offsprings here are coming at the F2 generation, and you see 3 of them are purple, even though they have different genotypes, and 1 of them is white. So 3:1. At this point now, we say that the generation, the distribution of the offspring is 3:1 phenotypic ratio, not genotypic ratio. Genotypic ratio will always be 1:1, and phenotypic 3:1. So an organism with 2 identical allele for a gene is called homozygous. I've talked about that. Homozygous, it is said to be homozygous for the gene controlling that character. An organism with 2 different allele of a gene is said to be heterozygous, and is said to be heterozygous for the gene controlling the character. Unlike homozygous, heterozygous are not true breeding. True breeding means it has to be either this. Now, I will put this here real quick. So, homozygous is going to be, this is homozygous. If you're looking at A and A, homozygous. This is also homozygous. So homozygous doesn't mean it has to be dominant. Homozygous simply means they have the same traits. The genes, the alleles are the same. Now, heterozygous is what you have. This is heterozygous, uppercase A and lowercase a. That will be heterozygous. So these guys are true breeding. These are also true breeding plants, or they are true breeding traits. They are true breeding. Heterozygous are not true breeding. So an organism trait do not always reveal its genetic composition. Okay, great. Great. So an organism trait do not always reveal its genetic composition. Therefore, we distinguish them between an organism phenotype or physical appearance and its genotype or genetic makeup. Like I told you, a purple flower that is heterozygous purple is different from a purple flower that is homozygous purple. All right. So in this example, flower of pea plant, PP, and homozygous purple and heterozygous purple plants have the same phenotype. They are all purple, but their genotypes are not the same. So if you look at this here, this is homozygous purple, of course, purple. This is heterozygous purple. This is also heterozygous purple. And now this is what we call genotype 1:1, whereas phenotype is 3:1. All right. So I'll stop the video here and continue the next one on another video.